Hello, welcome back, everyone. It's great to see you for the second day of the short course. Today, we will focus on how global environmental change contributes to pathogen emergence and dispersion, as well as host susceptibility. So we're very excited to have a set of terrific speakers lined up. And our first speaker will be Joseph Eisenberg from the University of Michigan. And Joe, are you okay to share your slides? And please introduce yourself. Just in terms of housekeeping, please keep your microphones muted, and you can post questions to the speakers in the chat, and we will gather the questions in the chat to address in the Q&A following the lecture. So we can go ahead and get started. The floor is yours, Joe.
Okay, great. Can you see my screen? Yeah, okay, thank you. So my name is Joe Eisenberg. I'm the professor and chair of epidemiology at the University of Michigan School of Public Health. And my area of interest is in understanding environmental determinants of infectious disease transmission in general. Most of my focus has been on enteric pathogens, diarrheal disease, and on vector-borne pathogens. I think some of the environmental issues of respiratory pathogens, which has not been as highlighted in the past, has been more highlighted in this COVID-19 pandemic. So I think it's apropos to be thinking about this now in this context about some of the environmental determinants of COVID-19 specifically and respiratory pathogens in general. So just an overview of my talk, which will be focused on talking about COVID-19 transmission in general, introducing this environmental transmission framework, a modeling framework, talking a little bit about dominant modes of transmission within COVID, and again, respiratory transmitted diseases in general, and then talk a little bit about wastewater-based epidemiology, which has been an ongoing interest in a variety of sectors, but has gotten increased interest in COVID-19 as of recent. So just to start off, say that coronaviruses are respiratory transmitted. They target the respiratory system. Most of the coronaviruses that are endemic cause what we call the common cold and are focus and target the upper respiratory system. So what's unique about COVID-19 is that it actually targets the lower respiratory system and therefore is inherently a much more severe disease and thereby resulting in the higher mortality that we're seeing in COVID-19 compared to many other coronaviruses that are endemic worldwide. So when we think and classify infectious disease pathogens broadly, we come up with these different kinds of classifications, such as directly transmitted diseases that move from person to person. The classic one that we think about, classic set of diseases, are sexually transmitted diseases, where in fact there really is no environmental phase of the pathogen. We've got vector-borne pathogens, we've got environmentally mediated pathogens, and we have zoonotic pathogens. So all of these, and many of these, the environment plays a huge role in transmission. Respiratory diseases have generally been classified as directly transmitted, measles and influenza and such, but and therefore the environmental process is often not considered for respiratory disease. But in many of these diseases, and even for diseases like TB, we've seen that the environment can actually play a significant role, and maybe it should be more classified in this environmental mediated category down here, as opposed to the directly transmitted category. But of course, that depends on scale what drives COVID-19 transmission, and again, respiratory pathogen transmission in general. At a global scale, transportation networks, as we have seen in the early parts of the pandemic, played a tremendous role in the spread of this pathogen around the world globally. And even regionally, we've seen many different pandemics, such as historically cholera, where transportation networks play a big role in moving and spreading disease. But at the local scale, the environment plays a much more important role for respiratory diseases. And many of our non-pharmaceutical interventions that we think about, wearing masks, distancing, cleaning surfaces, are environmentally mediated interventions. So the environment is an important part of transmission. And this diagram here provides a nice pictorial on the different types and environmental pathways that do play a role in transmission for COVID-19. And it's important here to realize and understand that viruses adhere to particles of different sizes, and it's those particles that move the virus through the environment. And so the size of the particle that's released, it becomes important with respect to the mode of transmission. So when someone sneezes, coughs, sings, or even talks, they release particles of different sizes. And some of the large particles depicted here fall to surfaces very quickly. And often we think of these, and if they fall on tabletops or doorknobs or different surfaces, that can promote a fomite-mediated transmission where somebody touches that contaminated surface, touches their eyes, nose, or mouth, membranes that are routes of exposure for the pathogen, and transmission is possible. You've got these medium-sized particles. That's where we see where they stay in the air for a certain amount of time and then drop off. It's where the droplet mode of transmission is much more important. If somebody is talking or sneezing in close proximity to somebody else, that the projectiles of those pathogens can result in exposure. And then for the smallest particles, here it says less than 5 microns. Some people say less than 10 microns. We've got the particles that stay aerosolized for quite a while, hours in the air, and that's where longer range transmission is possible. Talk about restaurants or indoor venues where you might not be in close proximity to somebody, but depending on the ventilation and airflow, can be exposed. So we've got different types of environmental pathways that we're going to explore a bit more later. Did want to talk a little bit about transmission and get into some of maybe transmission modeling that some of you may have already seen yesterday in the talk on transmission models, but I wanted to introduce this in a slightly different context and introduce environmental mediation in these models. So here's a depiction of how diseases spread with respect to what we consider the reproductive number R0. So with an R0 of 2, reproductive number of 2, that means that if somebody is infected and infectious, during their period of being infectious, they will ideally, if in a fully susceptible population, transmit to 2 other people. And those 2 will transmit to other 2. And you can see from this geometric progression, if each of these generation times takes a week, over 4 weeks, you go from 1 person to 16. One can imagine the next 4 weeks, you're in the thousands, and it doesn't take much time before you're into tens and hundreds of thousands of people. So this kind of depicts how you get this geometric, or what we've called exponential increase in cases due to the ability of one person to transmit to others. But in the real world, transmission doesn't happen nearly as regularly. So here's an example, and this is actually data from the SARS pandemic from the early 2000s, where one person, person A, infects 33 other people. And we see most of those 33, a big chunk of those 33 people don't transmit to anybody. But we see one person here, D, who gets infected from A, transmits to 10 other people. And here G transmits to one other person who then transmits to 12. So this kind of super spreading or heterogeneous method of spreading is really how diseases spread, because of the way in which, and depending on the way in which our social structure is defined. But the environment can often play a huge role in this heterogeneous spread. And an example I like to give is with norovirus. A food handler who's infected with norovirus, they actually you can be infectious after you've recovered from your symptoms, can easily spread to hundreds of people if they go back to work and handle food that other people are going to be exposed to. So environment can play a big role. Same thing with aerosol spread in this heterogeneous spread. So when we think about modeling transmission, and again, this is probably material that you've seen yesterday, but just to go over quickly to talk about how we think about the spread in the context of transmission models. Early 1900s, Sir Ronald Ross develops this idea from germ theory, this theory of mass action, that says susceptible people are exposed by contact from other infected people. So an infected person contacts a susceptible person, that susceptible person becomes infected. That's the basic framing of this. And that they will, in this model, a susceptible, we call this an SIS model, that the person that's infected recover and become susceptible again. So an extension to this model is what was developed in the late 1920s, is what we call the SIR model. That's the model that's most used in modeling diseases like COVID-19, where now we've got a susceptible person then can become infected when coming in direct contact with an infected individual. And that person will eventually recover from the transmission process. And when they recover, they actually have conferred immunity, and according to this model, long-term immunity. And so this is the basis of the kind of model that we'd be using in studying COVID-19 or other respiratory pathogens. To get into a little bit more detail about this model, the way in which new cases are generated, so I is the number of cases, and the IDT, just we would say, is the rate of generating new cases, is proportional to a transmission rate. And that transmission rate has two pieces to it. It has a social piece, which is the contact rate. And we think about the contact rate in the context of why we social distance. We social distance in order to reduce that contact rate. And then it's also a product of the probability of infection. That's the infectivity. So different pathogens and actually different variants may be more effective, have a greater infectivity. And so that's more of a biologic property of transmission, whereas the contact rate, more of a social aspect. And importantly, the rate is dependent on the number of infectious individuals and the number of susceptible individuals in the population. So the more infectious individuals there are, the higher the risk of getting new cases. That's why when we're in that exponential increase in cases, it becomes that much harder to control the disease. And then our reproductive number that we talked about before is just a product of the rate of generating new cases times the duration of being an individual being infectious. So how long somebody's infectious and the rate in which they're causing new cases during that time is basically our reproductive number that we talked about before. So one thing that's happening more and more is the introduction of an environmental state into this model. And that becomes important when interventions are environmentally based. Like, for instance, if we want to clean surfaces, we want to increase the, we want to kill off pathogens in the environment, basically. If we want to wear masks, we want to decrease that exposure of aerosol or droplet borne pathogens. So in this model, we've got the standard number of susceptible, number of infectious, number of immune that are part of the model, and we're keeping track of those individuals. And we've got this transmission rate. Right now, that in this case, the transmission rate is actually proportional to the pickup rate. That could be how much we inhale of a pathogen, which has to do with how much pathogen there is in the air, and also just our inhalation rate. And again, wearing masks can actually attenuate the amount that or prevent us from inhaling. We've got this pi, which is going to be our infectivity, just that we talked about before, and then E, which is the amount of pathogens that are in the environment. And then we got the recovery rate that we have talked about before, that people, after being infectious for a period of time, become immune. So importantly, as I mentioned, we've got the number of pathogens in the environment that we're explicitly accounting for. And we're accounting for the fact that there's pathogens will die off in the environment, or they'll leave the venue in which you're modeling. So it could actually, this mu could account for die-off, but also attenuation due to dilution, for instance. Again, individuals deposit and shed pathogens into the environment, and they pick up pathogens into the environment. So this provides a framework for us to think about explicitly the environmental interventions that we are interested in with respect to respiratory pathogens. And then our R naught now becomes a little bit more involved, but it the useful piece of this is that now it has properties of the environment, like shedding and pathogen persistence. So for instance, this term here, alpha over gamma, accounts for the pathogens deposited by infectious individuals during their contagious period. So, so these pathogens are shed into the environment. And then this term here represents the proportion of pathogens picked up by a susceptible person while the pathogens are still alive. So mu accounts for the fact that the pathogens are continually dying. And then pi, again, is that pathogen infectivity. How infectious is that pathogen? Again, as variants come in, pathogen properties of the pathogen are going to change over time. So this is our base model that we're and base kind of framework to think about how pathogens and respiratory pathogens particularly move through populations and are spread. When we think about, again, a respiratory pathogen, we think about a number of different environmental pathways that we may want to interrupt. So again, we've got the small particles that are aerosolized, and we may be interested in ventilation, improving ventilation to as an infrastructural change to actually minimize the impact of these aerosol particles. We've got the medium particles that are what we call droplet. They have droplet spread. They eventually settle onto the surfaces, but they are in the air for a short period of time. And then we've got the larger particles that settle much more quickly. And so this distribution of particles provides provides information about the fate of the different particles, and also the ability for a pathogen to actually survive on, like a virus, to actually survive on a small particle like this determines whether aerosol transmission is going to be important or not. Early on, we didn't think that COVID-19 could survive on these small particles. We thought that droplet spread was more important. Epidemiologic data was more suggestive that this aerosol aerosolized transmission actually was more important than we had previously thought. And so then these are various parameters that we need to quantify these transmission pathways and better model these transmission pathways, and therefore better explore intervention strategies. What is the shedding rate, the rate in which we're shedding? And that's for COVID-19, a function. Are people coughing? Are they talking? Are they singing? So it's going to be very behavior dependent, very dependent on venue. Shedding magnitude, again, how much virus you actually have in you, that could be a function of the pathogen, and the variants may actually have more, right? You might wind up having more virus that you're releasing. And then the concentration and how that varies over once in the environment. Inactivation rates, settling rates, touching rates, breathing rates, all these play a role in determining whether or not fomite transmission or droplet spread or aerosol spread is going to be more important. And that is the theme of what I'm going to be presenting to you here is, and the gist of it is that we often talk about, oh, is COVID aerosol spread, or is it more droplet spread, or is it, we thought it was more of a fomite spread pathogen before, and now we think that's less likely because it dies off quickly. But I think the message is that it's very context specific what mode of transmission, what environmental pathway is dominant. And I'm going to give just a few examples of that right here, to where, so when might droplet spread be dominant? Well, it might be a situation where density is high, contact is close, and ventilation is poor. Classic example might be a subway system or subway car. So in this situation, we might think droplet spread is going to be the dominant mode of transmission. Aerosolized spread may be dominant when contact is not close, but density is still high and ventilation is poor. Again, classic examples here might be a restaurant or an office building where you don't have to necessarily be that in close contact with somebody, but you're spending a lot of time in this one enclosed venue. Depending on the ventilation rate, transmission can be aerosolized spread can be a dominant mode of that transmission. Again, I think I mentioned this before. Can viruses be aerosolized? Do pathogens adhere and survive on small particles? That's a important part of whether or not, even in a given venue like this, the degree to which aerosolized transmission will be a dominant mode of transmission. And last, if we think about contexts in which fomite transmission may be dominant, a situation where self-inoculation rate is high, that's a behavioral kind of trait, and objects are shared. What comes to mind is a daycare center where kids are sharing lots of toys, they're touching their eyes, nose, and face a lot. Here we might think that dominant mode spread would be more dominant, even for a pathogen that might not survive readily in the environment, like COVID-19, like a lot of other respiratory pathogens. So, so the theme here is that context matters, and to determine dominant modes of transmission, we need to know about the host, information about the agent, information about the environment. And we talked a little bit about all these different features of the virus, of the host, and of the environment that could shift and the context in which we might think about one mode being dominant over the other, and thereby suggesting different mitigation strategies to address the spread in that particular venue. So I just wanted, the last part of this talk, I wanted to end to talk a little bit about wastewater surveillance as a potential powerful public health tool. It's become well publicized for COVID-19 because, as I'll talk about, COVID-19 is seen actually, or SARS-CoV-2, the virus is actually seen in and present in sewage. So just briefly, this type of surveillance can be quite useful in public health surveillance because it detects symptomatic and asymptomatic infections. So the presence of the virus in the sewage could indicate both symptomatic and asymptomatic transmission. It also is an early detection. We can detect, as I'll show a little later, detect rises in sewage concentrations of pathogens earlier and before you would actually detect it through case reporting. So there's been extensive work done on this with respect to polio. We've done some work, and I'll present some work with respect to this in the context of an epidemic that occurred in Israel. Extensive work on polio and other enteric pathogens. But as I mentioned before, the interest here is that SARS-CoV-2 can be shed in feces, even though it's not a transmission mode of transmission as opposed to the enteric pathogens. It has this potential for being an important surveillance tool. So just briefly, we were involved in analyzing some of the data from Israel. There was a silent outbreak in Israel where they detected wild polio in 2013 in their sewage. This is a huge, huge deal when we were at the brink of eradicating polio. Israel, a high-income country, shows sign of polio transmission. And these are just data that shows the wild polio at high levels early in 2013. This gray bar shows when Israel implemented OPV, the oral polio vaccine, and then we see a drop in the sewage. This was a second campaign here, a drop in the sewage levels of wild polio. And interestingly, and not surprisingly, we see this rise of oral polio, variety of oral polio virus, vaccine variety of the virus, rising at the beginning of the OPV campaign, and then a little bit of a rise after the second campaign, and then a drop. So we use these data and our transmission models to actually then predict and estimate the epidemic curve that have come through with respect to that we would predict happened in Israel during this time. Again, the important thing here is there was no clinical cases identified. So huge amount of transmission occurring of polio that was really only identified through this sewage surveillance. So if we think about it and apply this to COVID-19, we can see that we could be sampling at the wastewater treatment plant, and we can get aggregate information about epidemiologic patterns of transmission in a given target population that's served by this wastewater treatment plant, and get important information about seasonality and about early warnings of a potential increase in cases that might be occurring in this targeted population. And we could also increase our resolution and by collecting samples of sewage at trunk lines of the sewage network of sewage systems in order to pinpoint potentially where we might be seeing clusters of cases. The techniques here are molecular, they're highly sensitive, highly specific. I mean, we always are concerned about this viability issue because we are using molecular methods. We don't know if the DNA and RNA represent viable pathogens or not. So that's always an issue to keep in mind. But in these outbreak situations, this issue becomes less of an issue with respect to early warning system. And this gives an example here of the green lines, the blue lines are actually sewage levels of SARS-CoV-2 in the sewage, and the red is the case reporting. So we see that rises in the sewage precede the rises we see in the case reporting, and thereby this showing that this has a has a lot of potential with respect to providing good specific information about early detection of clusters of outbreaks in given regions. There's a lot of methodological development we need to do to make this really an effective public health tool. One question here is more virus due to, are we seeing more virus due to transmission, increased transmission, and thereby clusters of cases, or is it simply due to the fact we have more sewage? And that becomes really important when we're comparing from one target population to another across different treatment plants. The solution that has been found is that we need to normalize the virus data with a more common viruses that are transmitted through food, for instance, that really represent how much sewage there is in the system, and thereby, if we normalize our virus data with that, we get a much more accurate and less biased estimation of the temporal trends of transmission of SARS-CoV-2. And then the next is when we see viruses in the sewage, is that represent many shedding few viruses or few shedding many viruses? And so we really need to better understand the dynamics of shedding in humans, account for this time varying shedding rates in order to, again, provide a more accurate epidemiologic picture of what's happening in these regions. So I just want to end here by just kind of reiterating a couple of the key messages in this presentation. Many of the COVID-19 non-pharmaceutical interventions, those are not dealing with treatment or vaccines, are focused on the environment, and therefore a framework of thinking about environmental transmission becomes very important. And second, that because SARS-CoV-2 is shed in the feces, that wastewater surveillance can be and can enhance public health surveillance and add specificity in the way that we're really seeing for diseases like polio now, where it's become much more standard to be relying on the presence of wild polio in the sewage, as opposed to seeing clinical manifestations of polio, which would normally be acute flaccid paralysis, which is fairly rare. So this kind of wastewater surveillance can both be much more specific, it can and can pinpoint where transmission is occurring. It also provides this early warning system. So with that, I'm open to questions or comments.
Thanks so much, Joe. That was a fascinating presentation. There are some comments coming in here. Here's a question from Manolis. We were slow in identifying the importance of aerosols as a transmission route. So why was this? What was the sort of accepted thinking really that made the focus so much on fomites and droplets?
Yeah, that's a good question. I think part of this really shows that there have been people that focus on this research on understanding, what is the distribution of size of particles that are released, and what is the properties of viruses that are needed to survive on small particles that would be aerosolized. There are certainly people that are looking at that, but that has not been a sort of major thrust in the research. We really didn't, there's just lots of unknowns. Even with influenza, There were the fomite camp people that were convinced that fomites were important because you're getting higher doses, even if the infectivity is less, even if the survivor is less, but you get these high doses. There are the aerosol people say, well, even though you're getting lower dose from aerosol, you're inhaling more over time and it's more infectious, so that's more important. And so there are these really scientific debates happening, and so I think when COVID-19 came in, it's in the midst of this, and there's questions about whether masks were effective. Lots of epidemiologic data suggests we're equivocal about, well, maybe masks aren't that important. That's the message from the studies with influenza. And people thought that droplet spread was really important. So that carried over with COVID-19, but then we were just getting a lot of epidemiologic data with this pandemic and seeing that there was transmission occurring in restaurants from people that just weren't in contact with each other. So this evidence was just mounting that aerosolized transmission was important. That was more epidemiologic than sort of physical and for kind of information and microbiologic information about what's happening in the environment. So I think we were learning over this period, and the science was kind of revising, their opinions about different aspects of transmission and what was dominant and not dominant. And I still think this idea that it's context dependent is not widely appreciated. I think people want to think, oh, it's aerosolized. Oh, it's not fomite, or either or kind of thing. But I think that context matters, and it depends on the context and the venue.
There's a second question about water surveillance, which you showed from the Israel example is very important. So this question is about at this point in time in the COVID pandemic, with rapid changes and active identification of cases, how much do you gain from the wastewater surveillance in sort of practical terms?
You're gaining a lot. I mean, clinical surveillance is always fraught with biases. It's always fraught with, well, how much testing are you doing? And we can see even now we're doing less and less testing as we're ramping up the vaccination campaign. And so our surveillance is always biased by how well we're doing on the testing and identifying, whereas the wastewater sewage is just presenting an aggregate view of what's happening, and what people are shedding. So it's going to be less biased. It still provides, an early warning. I think in the beginning of the pandemic, when we were only testing the most severe cases, that delay between when we might see a peak in the wastewater and we identified clinical was much larger. So it's less now, but still, if you can gain days or even a week on where a cluster might be and you can act quickly, that's a big advantage with respect to public health action. So I think wastewater sewage surveillance has a lot of potential. It has a lot of potential for enteroviruses and other pathogens also, not just for COVID-19. But I think it's an important underutilized public health surveillance tool, and also in the long run probably relatively cheap compared, given how you can use one wastewater treatment plant to actually look at what's happening. Let's say you're looking at seasonality. When is COVID-19 might be coming back up, or when we might see, a new season coming in, if it does become more seasonal in the future. And then once you see it in the wastewater treatment, you might want to go into the sewage system and the branch line to say, well, where exactly are we seeing it in this area? So I think it has a lot of value. And
so just to, there's another question on the wastewater Surveillance. And this is coming from someone who's running wastewater surveillance in Bangladesh. And so the question was, what about in the context where you don't have a sewered system per se, so an unsewered sewage network? And do you have any advice to rule out how the asymptomatic and clinically tested patient ratio?
Yes, this becomes much more challenging in an area if there's no sewage system at all. And I think that was reflective in our work in Israel too, and because most of the transmission was happening in the Bedouin communities. A lot of Bedouin communities don't have, and so we don't have sewer systems. So we were seeing the data in the Bedouin communities that were actually had the most sophisticated sanitation infrastructure, and not really seeing it in the others. I think that in these other communities that don't have the sewage system, I think that you can do the environmental surveillance. It's just not nearly as efficient. But if there's evidence that that you're seeing a lot of transmission, there might be ways. I mean, it depends on the places where often there's informal canals that are moving wastewater through a community or downstream or what. So there are probably ways, depending on the situation, to take advantage of this system. Like actually Mexico City, we've worked in a little bit where there's all of Mexico City's waste goes to the northern Mezquital Valley in these canals. There's no treatment plant or there's no really system, but they're just these canals is where all the water gathers, and it's a point in which you can actually monitor the environment. So it is much more challenging though when you don't have a formalized system. So this system has mostly been used in areas where there is some sort of sewage system, more of an urban setting.
So there are several more questions, but for the sake of time, I think we'll have to move to the next speaker, but we will copy these questions over to the Slack, and hopefully you can answer them later, or at least other participants can continue to discuss them. So thanks so much, Joe. And we'll now move on to the next speaker, which is Kris Murray.
Hey there, Catherine, how are you going? Hi all.
Hello.
Coming through, and I'm going to share my screen here. Hopefully this comes through okay. All right, how's that look? Okay. Thanks a lot for the invitation to come and join this this really cool workshop. I'm really flattered to be asked to give a session on the link between COVID-19, disease emergence more generally, to ecosystems and environmental change. To give a little bit of a background on myself, I'm an ecologist, originally from Australia. I now work across two institutions, one in the UK, Imperial College London, and the other one is at the MRC unit in the Gambia, which is a branch of, now a branch of London School of Hygiene and Tropical Medicine. So I'm physically based in West Africa now. And here I lead a program, setting up a program really on planetary health or environmental change and health to sort of reflect the growing interest in this area, amongst high level health funders, I suppose. My background's a little bit diverse. I've just got one slide on this, just to sort of note that I'm coming at this topic very much from the ecological perspective. I'm trained as an ecologist. I did my PhD on diseases in wildlife, actually. And since that time, have sort of used that background to think about the way that pathogens move from wildlife into human populations. And so most of what I'm going to be talking about is very much this sort of this focus on the ecology of disease emergence. So just to sort of go back to, I'm sure you had lots of introductory material on this stuff, but just to sort of like put it straight out there that in comparison to the field that I'm in, which is conservation biology and environmental metrics and things like that, the metrics for health are really, really good globally. I mean, there's never been a time in history where the world's been living longer, you know, and it's a really positive success story here for global health, I think, over the last sort of 50 years or so. The inset graph here is just showing the life expectancy for all the countries in the world, and you can kind of see almost universally that we're living longer. Some of that is living longer with miserable conditions, but for the most part, you know, health metrics are pretty good. And particularly in comparison to the environmental metrics. This larger image here is a breakdown of the global burden of disease, and in blue we have the share of non-communicable diseases that are killing people and causing loss of good health. And you can kind of see this wedge is kind of growing through time, and that's really because it's sort of displacing this red wedge, which is here labeled other, but this is primarily infectious diseases here. And so you can see that like controlling infectious diseases has been one of those things that's really been one of the sort of major success stories in global health. But that's not to say that everything's sunny and rosy. And in fact, if you do extrapolate this curve, you might predict that, you know, infections go extinct more or less within maybe half a century. But of course, there are these sort of subsets or subcategories of diseases that are more problematic, more stubborn, more unpredictable, sometimes very impactful. I've just pulled up a couple of plots here of some diseases that are shown to have an increasing number of cases over the time series in this big data set called Gideon. So here we have plague, rabies, Lyme disease, and hantavirus, and these plots just show that they're increasing through time. This coming out of a study by Sebastian Funk et al. A few years ago. So that kind of illustrates that there are some diseases that are just doing things that we don't really want them to do. And that's sort of part of this category of diseases that we now commonly refer to as emerging infectious diseases. And emerging infectious diseases are those diseases that are appearing for the first time, or they're increasing in their incidence or their impact, or they're switching hosts, or they're doing things that is sort of unsettling us from a control perspective. And of course, all pandemics at some point are emerging. And here on the left-hand side, I've got just a couple of examples of the big pandemics, and I'm sure everybody's familiar with this story. And of course, this is the sort of thing that inspires Hollywood movies and books and all the rest of it. The important thing to note about emerging infections as a group, as a sort of a poorly defined, but a defined group nonetheless, is that the majority of them are zoonotic. And by zoonotic, I mean they're transmitted from another vertebrate host to people. And that includes both the species that we live with and that we've domesticated and that we farm, livestock, but it also includes wildlife at large. And wildlife actually accounts for around about 40% of all of these EIDs, the origins of these EIDs. And about 70 or 75% of all the zoonotic EIDs are coming from wildlife. And this is where this story connects with SARS, obviously, with SARS-2, obviously, that's where what this workshop's really all about, coronavirus 19 and this SARS-CoV-2 virus is the sort of contemporary pandemic. It overlays other pandemics that we're still tackling, like HIV. But this is sort of like become a bit of a classic story now. This is another wildlife origin zoonosis. The coronavirus comes from a family that is associated with wild bats, and in this case, in particular, in the genus Rhinolophus, or what we might call horseshoe bats. So what I'm going to talk about for the rest of the talk is really how this process of environmental change and the link to zoonotic reservoirs and zoonoses is related to this story around COVID-19. So before I kind of dig into anything, I wanted to sort of go back to some of the basic epidemiology of emerging infections. And in this case, it's particularly focusing on spillover. Spillover is that initial process of a pathogen moving from a wildlife host or a livestock host into another host. And in this case, it's into humans. And Lloyd Smith et al. Wrote this really nice paper about a decade ago now, sort of breaking this force of infection component, which is exactly the same thing that Joseph was just talking about before with respect to the transmission dynamics of diseases in general. and broken out the sort of the different components that are contributing to this spillover force of infection. And the three sort of take-home messages here is that the take-home message here is that there's three sort of broad categories of things that we think about when we think about spillover risk. The first one is the prevalence of a pathogen in the reservoir host, and that's affected by a whole bunch of things. The second is the reservoir human contact rate, and again, that is affected by a whole bunch of things. And then finally, we have the probability of infection given contact. And again, this is exactly the same stuff that Joseph was talking about before. And again, we see that step of the spillover process can vary by an enormous number of things as well. So the question for us is really, how do anthropogenic environmental impacts like land use and climate influence some of the levers in these three sort of pillars of spillover epidemiology? And then one thing that I think sort of separates this topic a little bit from COVID-19 in particular and other pandemics is that this is really just the initial step in the pipeline of disease emergence and onwards to pandemics. And so where we think certain things are really important for the spillover process, other things may be more important for the global spread to turn that thing into a pandemic. And it's already been mentioned, for example, the global travel and trade effect of spreading around a novel pathogen that has managed already to achieve human-to-human transmission. In some cases, land use and climate are facilitating the spread by changing the suitability of the environment into which the pathogen can be introduced. But more or less, I'm going to be talking mainly about this sort of spillover component and leave the global travel and trade side of the story to other speakers. So let's sort of get a really basic conceptual model here of a landscape. The landscape here is comprised of, broadly speaking, the biosphere, which is everything that's living around us. And within that, we have these other sort of circulating spheres. One of them is the domestic landscape, where we live, where we have our livestock, where we farm. And then we have this sort of other component of the biosphere, which is where wild animals are living. And of course, there's plenty of contact and overlap at these ecosystem interfaces. We are part of the ecosystem. We're having contact with wildlife all the time. Our livestock is having contact with wildlife all the time. And it's this sort of, melange of systems that are giving rise to some of the diseases that we are now sort of increasingly linking to in particular here, land use change. So some examples that I've got here, some pathogens that have moved from wildlife into humans include HIV, Ebola, rabies, and more recently SARS and MERS coronaviruses. Some of the diseases are sort of more associated with the disturbance of these systems, and that could include yellow fever, Lyme disease, and hantavirus. And then there are other diseases that also include this domestic animal or livestock component, so something like Nipah virus, which is transmitted from megabats through pigs into people. So these examples are sort of being collated in a number of different ways, but one of the ways is in this sort of like fairly narrative way, I would say. There's not a huge amount of quantitative stuff on this overall kind of picture of the number of diseases associated with land use change, but the hypothesis has emerged that where we go and interrupt landscape, where we go and have impacts in landscapes, in some cases that can catalyze the emergence or the increase of a new disease, the emergence of a new disease or the increase of existing diseases. And this is sort of broadly been phrased by a couple of different authors as the unhealthy landscapes or the pathogenic landscapes or landscape immunity type hypotheses. So I'm going to just sort of try and break that down a little bit. Here's another sort of, again, very conceptual model about the components that are comprised in a disease system, from the physical geography at the bottom of this plot through to the way that shapes the abiotic components of the environment, things like microclimates, soils and that kind of thing. Got a couple of layers in here for the vectors and the reservoirs which move pathogens around, and then we have a human layer in here, and it's this sort of, again, this is the sort of represents this biosphere that was really sort of illustrated in that previous plot. Then we have some kind of layer here which represents the human layer, and there's really, I see two major components to that. One is that we're constantly fighting diseases, and the other one is that we're variably looking for diseases. So we have this kind of management slice and this observation slice, which goes to complicate the way that we that we're able to measure and map different diseases. And at the top here, I've got, just 3, dummy diseases overlap to represent some kind of disease assemblage that might be sort of present in this particular landscape. This is what we might represent as a natural landscape, and the hypothesis around unhealthy or pathogenic landscapes is that we start to mess up some of these components in the system, in the environment layer in particular, through land use and climate change. That can change the number of reservoirs or vectors or their abundance. That can have impacts on the prevalence of the pathogen. And in some cases, it also affects the way that humans are utilising the landscape. And combined, that goes through this sort of process to make this landscape more diseasey than it was perhaps previously. So, and again, I should just say that this is very much the realm of hypothesis, and a huge part of the scientific effort in this area is about trying to sort of verify and validate the generality of this process. The alternative to that, well, before I get to the alternative, this is the slide I showed you before, and we can see this process through these specific examples. And I think that's where most of this theory really comes from mostly emerging and endemic diseases that we already know about. But occasionally there are entirely new, novel diseases that we didn't know existed and pop into the human population, and we start to connect the dots and think about the way that our own footprint on the planet has contributed to their emergence. So recent examples include Sars and MERS, and of course this whole area of science has been very noisy about the potential role of these things in the emergence of SARS-2. However, it's not always clear, in fact I would say it's most often not clear, whether there is some net increase in risk, a cumulative increase, where the overall landscape itself has become riskier than it was previously. And I think this again is like the topic of a lot of people's research. Here, this is represented by these processes variably impacting the host and the reservoirs of the environment, variably impacting the community of pathogens and the prevalence of those pathogens in this process. And combined, we really don't have a very good handle on whether that creates a net increase in risk at the landscape level. However, there are a couple of studies that have shown that there is some reason to think that this process could lead to a general increase in risk, or some component of the risk, well, what we might call here a risk triad, which comprises these 3 pretty distinct components of hazard, exposure, and vulnerability. So if we think about hazard, it might just be the presence of a zoonotic reservoir host in this landscape. So this study by Rory Gibb et al. That came out last year, a really fantastic study. What it shows is that through this huge global meta-analysis, is that in landscapes that are natural, you tend to have a limited difference between the abundance of zoonotic reservoir hosts and non-hosts. However, when you move along the x-axis here, you're moving into increasingly disturbed habitats. And what we see in these data is that the proportion or the abundance of zoonotic reservoir hosts increases relative to non-hosts. And so this is one plausible explanation for a generic increase in risk, although this is not measured as an outcome in humans. Another example is this study from Halliday et al. from Ecology Letters, also last year, and this shows again what we would consider to be some kind of a dilution effect of biodiversity. Here, anything below this dotted line represents lower disease risk in the presence of more biodiversity. And in particular, these authors isolated that effect as being attributable to the loss of biodiversity. So if something about the loss of biodiversity is selecting environments to be more parasitic, more pathogenic in the hosts that are left. And this again is not a human example, but the idea is that this could be one of the things that explains why we see this increase in emergent infections. And then finally, we have this sort of, this possibility that actually we can go into a certain extent and mess up the landscape, but maybe we're actually making these landscapes even healthier than they were before. An example of that is that a lot of health metrics in urban environments look a lot better than they do in rural environments. And that could be because we have this sort of combined effect of knocking out hosts and reservoirs, vectors and reservoirs, reducing pathogen prevalence in some cases, but probably more importantly, we've got this very important layer here of interventions and closer access to health infrastructure and that sort of thing. So it's possible that this land use change process, when run right through to that kind of urban endpoint, could be creating healthier from the perspective of zoonotic disease emergence. We just don't have a very clear view on this at the moment. One thing that I think is really complicated about this story is that if we try to build up the picture disease by disease, which is sort of what I was showing in those previous examples that have been really associated with land use change, is that we just have a fairly dizzying array of possible systems to deal with when it comes to human pathogens. So this is some stuff that came out of a study from 2015. There's this list of pathogens that have been associated with humans. This is kind of like a catalogue or an encyclopedia of pathogens. This gets updated every now and then. So previously, 10 years ago, 15 years ago, it was, maybe half the number of pathogens. But the point is there's a couple of thousand pathogens at least that infect humans across these broad pathogen domains of viruses, bacteria, fungi, and etc. And when we look at the diseases, the composition of the transmission sources of human diseases, what we see is that, again, here, the majority are coming from animal reservoirs, a bunch of vector-borne, a bunch of human-to-human transmission, and then there's sort of a subset that is sort of directly transmitted from the environment. Now, the interesting thing here is that almost all of these animal origin diseases, these zoonotic diseases, have multiple species involved in their transmission cycles. That's the grey bar here. A very, very small number is just a single host that we know about. The same occurs for vectors. Most vector-borne diseases have multiple vector hosts. Only a few of them have a single vector host that we know about. And then the other thing is that most of these, or a big chunk of these animal reservoir diseases, zoonotic diseases, are also transmitted by vectors. And a very large proportion of vector-borne diseases also have some animal reservoir host. So you can start to sort of see the complexity of these systems if you sort of imagine what is required for these pathogens to transmit or to complete their transmission cycles. The next thing that's really relevant to disease emergence, of course, is that we don't know about most of the possible diseases or pathogens that could emerge into the human population. For sure, nature is the ultimate reservoir for most of these things. We can go and we can test wildlife and catalogue all of the nasty viruses that they might have. But at the end of the day, it's really pretty much guesswork how many possible pathogens are out there that could spill over into humans at some point. Initial estimates of some work that I was actually involved in a few years ago put that number at around about 500,000 to 1.5 million viruses in mammalian hosts. And then more recently, a much more reasonable estimate that took into account the sharing network of viruses between species suggests it might be around 10,000. So you can sort of see that uncertainty is just absolutely huge at the moment. The point is that the majority of human infectious diseases are very complex beasts to try to unpick. So trying to predict the net impact of land use change on the entire community of human pathogens is just a very, very difficult task to try and achieve. So one way that we can try to think about the more generic outcome of land use or land use change, or in fact any other process, is to look rather than bottom up through this conceptual model here, to look down. We look at the global picture of all of the pathogens, their distributions, their, where they are, what they do, all that kind of stuff, all at the same time, looking from sort of like a bird's eye view. And I've got a couple of examples of what that might look like. Here is a map of what we're calling here disease emergence. Here's a map of disease richness. So this is the number of different pathogens per country in every country on Earth. And here, this is the sort of regionalization of pathogens, how similar the pathogen communities are across geography. And you can kind of see that it's very Wallacean in this sense. You have sort of Australasian groups of disease, African groups of disease, and whatnot. So what can we learn from looking down from this sort of bird's eye view perspective? So here I'm only going to have time to talk about one of these analyses. I'm going to take the middle one, which was the zoonotic disease richness picture. This is what zoonotic disease richness looks like at the global level. You can see there's quite a lot of variation. There's no real clear trend that you can see just by looking at the map, but we can use statistical models to try to work out whether there's some things that are more or less associated with this total diseasiness of countries. So in this model, we've got these types of variables: tourism, environmental components, we've got climate components, we've got health and GDP and all this kind of stuff thrown into the one model to try and explain what is at the center of this picture, which is disease richness. So what the green arrows show is where you have a positive association with these predictors and disease richness, and where you have a red arrow, you have a negative association. So what this is saying is that we'll take the red arrow to start with, we have an association of increased health expenditure on decreased disease richness, or conversely, more diseases where there's less health spending. On the environmental side, we have some other interesting patterns here. So we have mammalian biodiversity is the #1 predictor of disease richness at this kind of global scale. But we also have direct effects of climate going into this picture. We have effects, biogeographic effects of area. And then on the non-environmental side, we have increases in population size and increases in the human connectivity network that also are associated with the increase in disease richness. And then these kind of arrows going this way are just sort of showing that these variables are not always independent. They're, in fact, there's lots of sort of relationships between the variables themselves that we need to consider if we're trying to sort of work out this endpoint. So if we look at that model and then think about some of the things that we've sort of done to the planet in the last, say, 100, 150 years, we've modified 50% of the Earth's ice-free land surface. We've raised sea levels by a couple of millimeters, and that's accelerating. We've acidified the oceans. We've increased the rate of biodiversity extinction by maybe 3 or 4 orders of magnitude. We've increased the global average temperature by more than a degree. There's been a 3-fold increase in global population just since 1950, and a 56-fold increase in international travel just since 1950. So if you think about those variables that were in this model, we've got a whole bunch of variables that are associated with us, and then a whole bunch of variables that we've got associated with the things that we're doing to the planet. So I've sort of broken that up in this really simplistic way here. Here we have human impacts with some control. So we can spend the amount of money we can control the amount of money we're spending on health. We can, to a certain extent, modulate connectivity and the risks associated with it. We are already starting to, put a brake on population growth. So there's some control over these factors. On this side, we're affecting these things as well through the activities that I just mentioned, but it's in a very, very uncontrolled way. So we're affecting the climate, but we don't really know how to stop it. We're causing the 6th mass extinction, where we're seeing unprecedented biodiversity loss. And we don't know how to stop it. The metrics are terrible. We've spent 50 or 70 odd years trying to stop biodiversity loss, and there are really very few success stories in the scheme of things. So this is the thing I think that's very unsettling about the story around disease emergence. So to bring that back in the last minute that I've got here to SARS coronavirus 2, yes, it has a likely wildlife origin, so it's a zoonosis. That is not enough necessarily to hand the processes of its emergence over to the environmental change field. We just simply don't have enough information of the mechanisms of the emergence and the relationship that we have to this presumable wildlife origin or wildlife reservoir. There could be intermediate reservoirs. We just have simply no information that we can use to link the emergence of SARS to deforestation, to agricultural expansion, to climate change, to any of the things that we're used to talking about. Of course, there's been a lot of speculation about the role of wildlife trade in the wet market, but again, the evidence is really circumstantial at this point as to what the role of wildlife trade in the emergence of COVID has been. And of course, there's a WHO team that's been investigating that over the last sort of couple of months, and will continue, as I read today in Nature, will continue that investigation. So to summarize the key points here that I want to get across, most human infectious diseases are zoonotic. That is actually both endemic and emerging diseases. Around about 40% of emerging diseases are zoonotic, with wildlife origins specifically. Zoonoses, the risk of zoonoses and the richness of zoonoses are shaped by this interaction between biodiversity, habitats, land use, climate. So this is natural and anthropogenic factors. The host and the pathogen traits combine with the environment and with those human, social, and socioeconomic factors to generate global disease diversity. And we might imagine that that's correlated at some level with the overall level of risk from infection. And I think critically here, the global scale of disease diversity is controlled by factors that we affect, but we don't control, and we don't really understand what the implications of those levers that we're pulling in the global environmental system are really having on disease risks. Finally, all pandemics are EIDs, of course, by definition, but very few EIDs are pandemics. And since most of what we know about, this process of spillover and emergence is around the whole group of EIDs, it's very difficult to say what one specific pathogen, how one specific pathogen sort of fits the narrative here without sort of closer assessment. And for that reason, that's why at the moment it's still too early to tell what the role of environmental change has been in the emergence of SARS-CoV-2. Got the 2 in the wrong spot there, sorry about that. And then I guess this is the topic of the rest of the workshop. That doesn't mean that it's not too early to leverage COVID-19 and the global responses and impacts that it's had to think about how we might emerge from this global health crisis in a better position to combat future health crises. And a lot of those things are these big systemic, really sort of existential threats like climate change and land use change, which I think are in the long term represent the biggest global health threats that we're facing at the moment. I'll leave it there. Thanks so much for having me.
Thanks, Kris. That was really an excellent presentation and very, lots of thought-provoking material there. We're waiting for some questions to come in. In the meantime, can you maybe comment on what the sort of, there are a lot of uncertainties here that you've mentioned, and what some of the sort of key priority research gaps are, or sort of where the opportunities are from a research point of view to start sort of filling in some of these gaps in knowledge.
Yeah, sure. I mean, I think from the perspective of this idea that we're creating landscapes around us by our own impacts that are in some way fundamentally more or less risky. This suggestion has been around for a really long time. It was in about 2000, I think, when the first sort of paper around this sort of pathogenic landscapes or unhealthy landscapes idea came out. And the thing that sort of struck me over the past 12 months with the emergence of COVID is just how unreasonably dependent we are as a scientific community on borrowing the information from these sort of case studies to make general assessments about the role of environmental impacts on disease emergence and pandemic risk. I think there's a huge gap between those two things. And that hasn't stopped, of course, a lot of people sort of jumping to conclusions. For example, we have to shut down the wildlife trade in China. I'm not saying we don't need to do that. I'm just saying that we have absolutely no idea what the sort of, the full picture of implications for such a move might be. And in particular, we don't really have much of an idea of how that would reduce or eliminate pandemic risk. And I think that's the key bit. A lot of the processes are so complicated, and there's so many different pathways of disease emergence that jumping to, particularly strong, policy positions may not really have the desired effect at the end of the day. I think there are a lot of other reasons to do things like regulate the wildlife trade more effectively, particularly from a conservation perspective. But the evidence around this kind of this particular sort of nexus issue of environmental change fundamentally, or land use change in particular, fundamentally creating unhealthy landscapes, I think a lot of science needs to fill in a lot of that gaps. An example of that is that increasingly over the last 20 years, there's been this sort of movement of trying to quantify the ecosystem services in landscapes around us so that we have some way of, doing simple cost benefit analysis about the things that we do, develop a hotel on the beach, whatever, create a dam in this reservoir basin. And we can do things like, estimate the sort of value of biodiversity, or put values on the water filtration services of healthy forests and things like that. We are miles away from being able to do that same thing for pandemic risk. We are miles away from connecting specific interventions in landscapes to the overall reduction in pandemic risk. I think there's some things that obviously from a precautionary principle make a lot of sense, but other things I think could be giving rise to more harm than good. And I think that for me is where the COVID-19 global health crisis has revealed a lot of the cracks in the epidemiology of emerging infections.
So there's a related question, which was about, you mentioned the precautionary principle and things we can, things that make sense to do based on the precautionary principle in light of these uncertainties. Can you comment on what some of those are?
Yeah, I mean, I've been increasingly evasive on this question. And I should say that, before COVID emerged, I was perhaps, more on the side of precautionary principle being the thing that we should invoke straight up for, that it's somehow stopping everything that we're doing is the way to combat this risk. And since that time, I've kind of thought, well, really to do that effectively, you would have to, and what people are really talking about is eliminating spillover. Okay, so eliminating the very first step of this change. This is what people are sort of saying, stopping pandemic at source. So there are some things that could stop pandemics at source. That could be, for example, preventing human-animal contact. Can we prevent human-animal contact realistically, in the world? I don't think we can. I think there are ways that we can make our interactions with nature slightly, less risky from a disease transmission perspective. But it would be a massive loss for the planet if we all of a sudden had a global retreat from the interface that we have with the environment. I think that's a disaster. There's a lot of evidence to sort of show the benefits of nature for people. So if we start advocating we need to eliminate spillover, in that case, we really do need to eliminate the interactions we're having with wildlife and with livestock and our livestock with wildlife. I think that challenge is really too big to get on board with at the moment, given our understanding of the sort of, the hidden costs of something like that. I think that's a really bad idea to advocate for that sort of a thing. So, again, I don't want to be sort of roped into saying any specific thing. I'll go back to the wildlife trade example. There are lots of really, really excellent reasons to regulate and make it better, and in some cases ban it. Most of those reasons for me come from a conservation perspective, and the pandemic risk argument component of that doesn't stack up for me, at least. So that I would just leave it at that sort of as a sort of a simple example of how complex this issue of response and stopping pandemic at source really is.
Oh, that's very interesting. And, it was very interesting to hear you comment on the evidence that's available regarding the health, the benefits, many of which are health benefits of contact with nature, which is a very active area of research among ISEE members. And so how, with Sars, something I've struggled with is, how does this all sort of fit together? Because we're hearing many of these suggestions to limit nature contact where, many of us are working on research related to the health benefits of nature contact. So some interesting paradoxes there. So there is another question here from Manolis, and on Tuesday he gave an estimate of about 70% for the proportion of emerging infectious diseases that were due to zoonosis. And he was asked whether this percentage referred to all new diseases or only new infectious diseases. And maybe you can comment that. I think what he responded that practically all emerging diseases are infectious. And
I can clarify the stats and the source of this stat. It does refer to infectious diseases. There's a paper by Kate Jones in 2008 that catalogued these things that we call emerging infectious disease events. There were about 330 or 40 diseases in that database. Not all of them were independent, so there were multiple examples of the same type of disease, what we call disease event. And it's 70% of those events that have that are considered zoonotic, and so it doesn't include other emerging health health issues. The interesting thing about that stat is that it's probably one of the most misused stats when it comes to talking about emerging infections, because then, of course, you see, well, 70% of emerging infections come from wildlife. That's not true. Actually, less than half of emerging infections come from wildlife. And we're not even talking about emerging infections. We're talking about emerging infectious disease events. So again, that could be plague on, you know, in Senegal, in Yemen, in America would count 3 times in that database of only 335 diseases. So there's a few issues with that. That database is out of date. It is consistent with other estimates of these stats. For sure, the majority of infections and emerging infections are zoonotic. But yeah, we do need to be a little bit careful with the way that we sort of interpret the stats.
So then I think we have time for one more question. And there's a question here about hotspots for research prioritization, which I guess you've sort of commented on about the knowledge gaps. And I don't know if you have anything else you'd like to add. And there's a specific paper here that was someone has asked you to comment on, but I think what we can do there is put it in the Slack channel, and you can have a sort of exchange about the paper in the Slack channel, where I think
Sure.
is a better forum for exchange on this. But just to say thank you, that was really fascinating, and I would really love to see more interaction between this sort of ecology and the environmental epidemiology communities, because I think certainly environmental epidemiology has a lot to learn from the kinds of work that you and your colleagues in ecology are doing. So thanks so much, and we have a 10-minute break now, and then we'll come back and hear from Jan Semenza. Welcome back. And I think we can move on to the next presentation. Jan, the floor is yours.
Thanks, Catherine. Let me see if I can share my screen. See if I can arrange this. I can't see the screen here. Oops. Is that okay?
Looks good to me. So.
Good. Great, thanks, Catherine, for organizing this event. Thanks for inviting me. It's been very exciting so far. I would like to present some work we have done at the European CDC. In fact, here you see an image of the European Centers for Disease Prevention and Control. We are located in Stockholm. We are a European agency, so we report to the European Commission, and our mandate is restricted to infectious diseases only. We are much smaller than the US CDC, for example. We have a staff of like 300 individuals. Only a small part of those are technical experts, maybe 60 or so. However, our budget has now been increased for 60 million to maybe double due to the COVID pandemic. And we have obviously been involved in the response to the COVID pandemic very intensely over the last year. But basically, initially our mandate was to look at infectious diseases when it comes to from a surveillance perspective, to monitor all reportable diseases, and then conduct scientific assessments, epidemic intelligence, and response to potential outbreaks. And as I say, we are a small agency with a small staff, so you can't really compare us to the other CDCs in China or the US or in other places. But let me see. The question I would like to address here in this talk today is, what are these infectious disease threat events that we are concerned about right now in the world? What are these epidemics? And what leads to these type of epidemics, similar to what Kris alluded to earlier? What are the underlying drivers of these events? And is it potentially possible to monitor those drivers to prevent infectious disease threat events in order to build robustness and resilience in the public health system in Europe for these events? And so these are the kind of questions we wanted to ask here. And we're not trying to say that we can predict these kind of black swan events that are highly unlikely but potentially catastrophic, like COVID-19 or HIV. But maybe we can have a handle on the emergence of endemic diseases, potentially, to see if there's a change in patterns and change in drivers that could potentially indicate an increase like that. But as Kris very nicely illustrated, it's very difficult to predict these type of events. So in order to try to approach and understand underlying drivers and potential scenarios that they cause, the best thing would be to go to Greece, to the Oracle in Delphi, and consult with the Oracle to see what the future holds. Unfortunately, you see that the Oracle here is kind of dilapidated right now, doesn't work as well as it used to. And we didn't really know what to do next because we didn't have this feature anymore. So we basically resorted to the next best things after the foresight, after the Oracle, and decided to conduct a foresight study. which is basically bringing a group of experts like yourself to the table and brainstorm with these individuals what potential drivers to these type of outbreaks could be, and taking the context into account. The European Union, as you know, is a hotspot for emerging infectious diseases, and it's interconnected and interdependent, for that matter, with the rest of the world, which means that it is very much a hotspot for these type of outbreaks. And then can we potentially anticipate future disease outbreaks? And this one here was done in 2008. So our goal was to try to anticipate the future by 2020, looking forward 16 years, 12 years or so at the time. And so we asked these experts to identify key infectious disease drivers, and we plotted them out in workshops into different groups and organized them according to different features. We then conducted a literature review to validate our expert assessment, and developed scenarios based on the plausibility of these type of events, and particularly to see whether there are, assess the severity of these type of events to European public health. And then we developed public health action, how every single event can potentially be countered with specific steps in the public health infrastructure to prevent the fact that we get potentially overrun as we have for COVID-19. So this is what we outlined in this paper with all the different actions that need to be taken. And we came up with a list of drivers that we grouped into 3 categories, here illustrated into globalization and environmental change, social demographic change, and public health system. And within those 3 categories, there are obviously a number of other drivers. For example, in the first category, we have environmental and climatic change, travel and tourism, migration and global trade, and we group those into global environmental change. Then in the second category, we have the social factors, so inequality, demographic change, prevention and treatment, and lifestyle. For example, vaccine hesitancy would be in this category, or use of processed foods, so the change of lifestyles over the years and things like that. And then the third category, the public health system, we are looking at surveillance and reporting, healthcare system structures, animal health and food safety, and research and development. And so we basically came up with these 3 different categories as potential drivers for infectious disease threat event in Europe, and asked ourselves, how can these drivers now interact in a synergistic or potentially antagonistic way to create scenarios or stop scenarios from happening? So can we foresee how they potentially interact? And again, we use this foresight approach, and this is what we came up with, and it's a very busy slide. And you don't need to look at all the details, but basically on the left side here, you have the key drivers, again, grouped into global environmental change, social demographic change, and public health systems, and the different subcategories, and then how those interact to create vector-borne disease outbreak, extensive drug-resistant bacteria, sexually transmitted infections, foodborne infections, resurgence of vaccine-preventable diseases, healthcare-associated infections, multi-drug resistant tuberculosis, and pandemic flu. And so these were the scenarios that we came up with based on our foresight approach. And as you know, foresights are very useful for strategy planning and priority setting and all that. But obviously the big question here is this true or not? Can we potentially validate that? What can we do to see whether this is actually true, or whether we just threw some scenarios out of the hat and this is in fact not true at all? And so we basically decided trying to validate by using ECDC data, because at the European Centers for Disease Prevention and Control, we conduct 2 types of monitorings. We have the disease monitoring that we are mandated by law, as I mentioned earlier, where we monitor something like 50 different types of diseases that all EU member states have to report to ECDC. And we have built the TESI system, the European surveillance system, where all these data are reported to. And we clean the data, tabulate them, and make them available to researchers like yourself for epidemiologic analysis, and in order to understand pattern and trends and things like that. And so that's one of the core activities that we do. We produce this annual epidemiologic surveillance report that we disseminate widely about the state of affairs when it comes to infectious diseases in Europe. However, we also conduct event monitoring at ECDC. And that's a different type of surveillance system where we monitor with Google searchers and other type of search engines. We use confidential databases for that matter. And we basically conduct this European surveillance, early warning and response system to monitor all kinds of events that are picked up by the media, for example, and we try to validate them, capture them, validate them, filter them, and then decide whether we need to bang the drums, alert public health, come up with a signal and alert to take public health actions and control measures. So these are the two monitoring systems that we have at ECDC. And we decided that since we were interested in the underlying drivers in these potential scenarios, why don't we compare what we had found against these against this database of epidemic events? And so this is the team at ECDC that that assesses these epidemic events on a daily basis in the Emergency Operations Center, illustrated here. We meet at 11:30 every day, and we discuss all these epidemic events that are going on, and we assess the threat to Europe. And if need be, we write a risk assessment or a threat assessment about a potential epidemic event in the world that could potentially impact European citizens or Europe at large. And all the data is entered into a database that we call the Threat Tracking Tool, or TTT, illustrated here. And we write up these communicable disease threat reports that we publish on a regular basis. But as I mentioned, we also produce these threat assessments and rapid risk assessments. And we basically decided to rely on this type of a database to analyze every single event that was of interest to us that we found in the database. And we extracted the underlying drivers for every single event by going through the communicable disease threat reports that had been written about the events, the threat assessment, rapid risk assessments, but also the peer review literature, the scientific articles, expert consultation, mission reports, everything that we could find, we extracted that was available in the gray literature or in the peer review literature for every single event. And on this slide here, you see the type of events that we identified. Threat event category: food and waterborne diseases, vector-borne diseases, zoonosis, vaccine preventable, multi-drug, healthcare associated infections, injection drug use associated infections, sexually transmitted infections, influenza, and airborne infections. And you can see examples here of what we usually pick up during these roundtable discussions and epidemic intelligence, as we call it. And so when we looked at this list here, we extracted the drivers for those individual events and came up with an assessment like this. So if you plot out the number of underlying drivers for every single event here, you see that these events very rarely are caused by one single individual driver, illustrated here on this in this bar graph here. The majority of the drivers are, in fact, the majority of the events are caused by multiple drivers, 2 drivers, 3, 4, 5, or 6 even. And you can see a number of these type of drivers contributing to these infectious disease threat events, grouped here by the categories that I just mentioned. So the take-home message here is that epidemic events are rarely caused by one driver, and we live in a multifactorial world, as as Kris very nicely illustrated. And we plot that out here based on empirical data from the epidemic intelligence data. And then we plotted out the drivers here at the top and the epidemic events at the bottom, and now it becomes entirely uninterpretable, right? I mean, this is very difficult to process. This is very dizzy and confusing to look at. Here at the top, you have the socio-demographic drivers, the public health system driver, and the global environmental drivers in those three categories with the sub-drivers, and again, the epidemic events at the bottom. But you can't really tease that out. So if we take a more strategic approach to group them in a multi-dimensional space where you have all the drivers in a multi-dimensional space against these epidemic events, you can create this kind of cluster dendrogram using this hierarchical cluster analysis that we conducted with Joachim Brock-Löw, who spoke the other day here as part of this meeting. And you can see that a number of these type of drivers cluster together, and you look here, for example, migration and social inequality. They occur often in concert and occur together and cause a certain epidemic event. Or we see climate and natural environment. They act together in a way to precipitate an event. But the one thing that stands out in this particular slide is the fact that travel and tourism is the most important driver of epidemic events in Europe. So travel and tourism stands out when you look at this dissimilarity distance and when you look at the frequency of contribution of travel and tourism to these type of epidemic events. So travel and tourism is the most contributor to epidemic events in Europe, illustrated figuratively here on the slide, where you can see air passenger volume coming into Europe from all over the world. And in fact, Europe, the European airspace is one of the busiest airspaces in the world. There are 600 million passengers arriving every year. And this is where individuals are coming from. So Europe is blacked out here because you have the origin of the international air passenger volume throughout the world coming into Europe. This is based on the IATA data, where they map all the passenger volumes throughout the world. And they're coming into Europe. I don't know if you can recognize the map of Europe here, but you can see the big airports in Paris and Madrid and London and Frankfurt highlighted as these bubbles of representing the passenger volume coming into Europe. It turns out that a lot of outbreaks that we have experienced in the past have in fact been introduced through air passengers that arrive in Europe in a viremic state, which means that they have the virus circulating in their blood, and then they're bitten by a local mosquito that precipitates an outbreak. For example, in 2012, there was a big dengue outbreak in Madeira because Aedes aegypti, which is an invasive species, is present on this island in Madeira, and there was a big outbreak of 2,168 dengue cases at the time. But in continental Europe, we have a different type of a mosquito, which is called Aedes albopictus, or the Asian tiger mosquito. And we have also seen outbreaks in continental Europe of dengue, but also of chikungunya, another tropical disease, also transmitted by Aedes albopictus. There's been a dengue outbreak in Croatia and then in Italy in 2007 and 2017. And so we basically decided to analyze the relationship between air passenger volume and these type of dengue outbreaks that I mentioned earlier. And so in order to get an assessment of dengue outbreaks throughout the world, we conducted epidemic intelligence. We extracted information from dengue maps and communicable disease threat reports to identify all the areas where dengue outbreaks had happened recently. Then we contacted the European surveillance institutes and reference laboratories in Europe to ask them about dengue importation cases that had been reported to the laboratories or these national surveillance institutes. And we used data from the International Air Transportation Association, or IATA, as I mentioned, to get a handle on the volume of international travelers. And then the presence of the Asian tiger mosquito using vector surveillance data that we conduct here at the CDC, but also at EFSA as part of the ViborNet project. And so these are the data that we tapped into in order to assess this relationship between dengue outbreaks and passenger volume. Is it possible to model the dengue risk using passenger volume? And here on this slide here, we see the destination of international air travelers coming from anywhere from the world into Europe by country. And so this is plotted out by month for all the EU member states, listed here by volume. And here you can see the passenger volume origin, so where dengue currently is present, and the passenger volume that's flying out into Europe from these destinations like Miami or Delhi or Bangkok. So these bubbles represent the volume of individuals coming into Europe from dengue active areas during July to September, and this was for 2010. And so then we look at international air traveler arrivals into Europe from dengue active areas. So before it was everybody combined, and now we just tease out from dengue active areas based on that map I just showed you. And then again, this one here is by month, and you can see that during the summer months, we see a spike in these type of passengers arriving from dengue active areas into Europe. And this is where people are flying into. And so this slide here on the left illustrates the passenger volume from dengue active areas into Europe. You can see the yellow area on the map here, which illustrates the area where Aedes albopictus was present at the time. And as you know, the Asian tiger mosquito has expanded since, and I'll show you some maps on that. But you can see these this high passenger volume into areas where there is a risk of transmission because the vector is present in those areas. We then conducted a multivariate analysis, excuse me, where we looked at the risk of dengue importation into Europe using this passenger volume data that I illustrated, that I mentioned before from IATA. And so we basically tried to model to see whether it was possible to predict the occurrence of dengue cases in Europe with the passenger volume coming from dengue active areas. And what you see on this slide here, travelers from dengue active areas, it turns out that the incidence rate ratio is the tallest, is statistically significant for August and September when these passengers are arriving, and you can see that the incidence rate ratio increases quite a bit. And this is exactly the time when the mosquitoes, the Asian tiger mosquito, is at its peak. And so this is when we see this type of transmission. A lot of people are coming in exactly at the time when mosquitoes can potentially spread the disease onwards to unexpected individuals. We also can analyze the risk at airports, and here we looked at the European airports and the passenger volume coming into these airports, and you can see that more than 50% of the risk arrives in 2 Italian airports where the Asian tiger mosquito is present, and at the time in 2010 that was Milan and Rome. And so this kind of an analysis allows us to provide this type of information to the public health practitioners in Italy to tell them when to implement surveillance, seasonal surveillance, and localized sentinel surveillance for these type of hotspots in order to contain an outbreak if need be. So in summary, this analysis found that the highest risk is restricted to these 3 months that I mentioned, and that's exactly when the vector density is the highest, and the risk is confined only to a few cities. And when we looked at the expansion of Aedes albopictus after 2010, just a few years in 2013, we had seen that 39% more travelers are projected to arrive in those areas, in part because more people arrive from dengue, because dengue is expanding worldwide, as you know, as well, but also because the risk in Europe has expanded because Aedes albopictus is now present in more areas in Europe than before. And this kind of empirical model allows us to develop a spatiotemporal element to early warning systems by telling public health practitioners when and where to look out for these type of outbreaks. The next driver that I would like to touch on is global trade. And here illustrated with this boat here, it turns out that Aedes albopictus has moved from continent to continent via trade and expanded tremendously throughout the world. It's an invasive species and a very aggressive biter and vector. And it was introduced in the 70s into Albania, and then in the 90s into Genoa and Venice in Italy. And the vector expanded ever since the introduction, because he found a very hospitable ecologic niche in the Mediterranean region. And these vector surveillance system that I mentioned earlier has now picked up Aedes albopictus in a number of NUTS 3 levels throughout the Mediterranean. But this one here is for 2008. If you look at 2012, we have seen an expansion of these areas throughout the Mediterranean. You can see it even present in Holland, where the mosquito is present in greenhouses, not outside, but in air conditioned or in climate controlled greenhouses. So this one is for 2012, and this one is 2020, and you can see here this massive expansion of Aedes albopictus throughout Europe and moving further north, particularly along transportation corridors. It's now also found in Germany and in Switzerland and close to Paris as well. We modeled the climatic suitability for this particular vector, for Aedes albopictus and Egypti. And on the right panel, you can see that the climatic suitability for Aedes albopictus is very high, and we can potentially even see it in Denmark and southern Sweden, maybe even southern Norway, based on this analysis here. It hasn't arrived there yet, but potentially this ecologic niche is available for Aedes albopictus. Whether it will be able to transmit these tropical diseases in those type of habitats remains to be seen, because it also needs to have a high population density in order to drive an epidemic. But we believe that the climatic suitability is ripe for a further expansion of this invasive species in Europe. And
Jan, 1 more minute, please.
Oh, okay, good. So, let me skip this, and I'll go right to, let me go to the COVID one. So the last example then is human-made environments, and here I would like to touch on the mobility patterns during the pandemic, and whether this kind of restricting mobility has impact on contact rates in preventing COVID-19, and whether that should be followed up with contact tracing and whatnot. And so we used, for this particular example, the Citymapper's mobility index. And here we cover the time, the month of March. And we compare that to a baseline in January, early January till to February 2nd as a baseline. So everything that's over that baseline will be 100%, but it can also be below that baseline. And we conducted a multivariate fixed effect model to look at the contribution of these measures and used the Oxford Government Response Stringency Index. And so these are the cities that we looked at with the Citymapper mobility data that's available. So this one here is for Europe, this one here is for North America, and then for Asia. And you can see here a dramatic decrease in mobility using this kind of Citymapper mobility data. So if you look at Milan at the bottom there, for example, you can see a relatively modest decrease because Milan had experienced a pandemic very early on and implemented a number of restrictions already, mobility restrictions, and so that's why that curve is pretty low. The Russian cities here, St. Petersburg and Moscow, they were not impacted so early by the pandemic and showed a later kind of a decrease in mobility. And then among the Western European cities, Stockholm stands out where I live because the government here had decided not to impose stringent measures, and our society has been relatively open, and you can see that this is reflected in the mobility index here. Then comparing North American cities, they show similar type of patterns. Seattle was impacted early on, similar to Milan actually, probably not as early, but again, it's a bit further down compared to the other cities. Mexico City shows weird fluctuations, which we can't quite explain, but we see similar trends here of the decline. And then if you look at for the rest of the world, the Australian cities coming down quite aggressively, whereas the Asian ones are more flat because they were impacted by the pandemic earlier on, like, as I mentioned earlier. And here we're looking at individual days mapped against the stringency index, policy government stringency index, and the mobility index, and you can see that the stronger the index is, desto less mobility there is. And when we run a multivariate analysis here, and obviously there's a lot of multivariate collinearity here, we do see effects of school closing, workplace closing, and canceling of public events and transport. And these have all impacts on the mobility in our model, whereas fiscal measure, which was our controlled monetary measures, did not. So we added these type of variables to assess the validity of our model, and they don't seem to have an impact on mobility as expected. In summary, understanding the effects of public mobility is very important to slow the spread of the pandemic. And we see these trends in mobility in relationship to different policy interventions. And the most important slide here, these are individuals that contributed to the work. I mentioned Joachim Rochler, but there are a number of other individuals that have contributed to that, particularly Bertrand Soudre and David Stuckler. And I'll leave it at that.
Thanks so much, John. I see one question in the chat here, which was related to one of the dengue analyses, which was why use January as the reference month in the multivariable analysis?
Because it was the lowest month that we chose that as a baseline.
So you showed some nice examples of how travel and tourism and trade contribute to some of these vector-borne diseases in Europe. How well is it understood, or how well have people been able to sort of attribute the spread of COVID to these types of drivers?
So, I mean, that's a good question. So let me backtrack for that. I mean, a few slides that I didn't show was, for example, for dengue. So we saw this relationship with air passenger volume, but ironically, during the COVID pandemic, we still saw some outbreaks of dengue and even chikungunya in Europe. So we believe that air passenger volume is important, but there are other factors as well. So it's a stochastic event, right? It's just a combination of things. So I think it's a contributing factor, but it's not only a factor. Then regarding the spread of COVID, SARS-CoV-2 for that matter, obviously the air passenger volume has contributed to the rapid spread of the pandemic, and we have seen the introduction into Europe as a result of air passenger volume. There's no doubt that there is a link with that as well.
Any other questions here?
So if we look at these drivers for these type of epidemic events, we compared our prediction with the real data, and we, in our foresight study, had in fact underestimated the contribution of global environmental change to epidemic events. 61% of these type of epidemic events in Europe are due to global environmental change. Travel and tourism, as I mentioned, is the most important one, but overall as a category, global environmental change is the most important one, contributing to 61% of these events. And so that's a big contribution. And we had underestimated that in our foresight study. So we did a comparison between what we had as considered plausible, what we estimated, and then the actual. And it was striking to see the difference, how we had underestimated the contribution of travel and tourism, but global environmental change in general.
It's very interesting, and you know, it will be interesting to see how that informs thinking going forward the next time you do a foresight.
Yeah.
So, well, for the sake of time, I suggest we move on. Thanks very much, Jan. And if there are any further questions, we can take them in either the chat here or in the Slack channel. And our final speaker today is Tara Sabo-Attwood from the University of Florida.
Having trouble sharing here. How about this? Does anybody see anything here?
We see your slides. They are not in presentation mode.
I can't even see them on my screen here, which is odd. Let me unshare here for a second. What about now?
I see your slides, but can you put them in presentation mode? Do you see? Let's see.
Very strange, because I can't see. Of course, I tested this morning here. Let me try to. I'm seeing like a very strange blank slide here. I'm not even seeing. Oh, here we go. Hang on. All right. Is it in the right mode or no still?
Yes, but they're small. We can see if it's easier. We can see if we can present the slides from here. Ariadna, is that a possibility?
So you don't see it now?
We see it, but it's very small.
Huh.
You should click on duplicate, you know, on that up there on the left, Tara, as you had it before.
Good grief. Try one more time here.
Okay, great.
Perfect.
That's it.
You
Perfect.
got it?
Yeah.
Oh, gosh. You know, of course, that's when you get all set up and try this 20 times and it works, and then it doesn't when you need it to. So my apologies. So today, I was asked to come and talk to you about environmental pollution and resilience to infections. I have somewhat of a background in this, so I'm Tara Sabo-Attwood. I'm a professor and chair of the Department of Environmental and Global Health at the University of Florida. I also serve as a Dean for Cultural Affairs and Wellness Program. So I wear many hats, but the hat I'm going to wear today is as an environmental molecular toxicologist. And so my background is really in trying to understand the relationship between exposure to pollutants and endpoint disease, and one of those endpoints being susceptibility to infections, which is a space I've been working in for about the last 10 years or so. So I'm kind of going to be talking about this through that lens today. And again, thank you to the organizers for inviting me to come talk on this topic today. So what I thought I would do was cover a couple different topics here. First, I want to talk a little bit in general about the concepts surrounding the interplay between pollution and infections, and then introduce just a few mechanisms of action really related to exposure and the immune system that I think are likely important in this space. I'll try to talk a little bit about what we know so far about these mechanisms in the context of pollution, SARS-CoV-2 and COVID-19, and then really summarize up by talking about some important gaps and ways we might move forward with research initiatives today. So really this idea that there's interplay between chemicals and microbes where they interact with each other, whether that's directly or indirectly to influence health outcomes, is not really a new idea per se. And historically, there are quite a few examples for this. So for example, in the 1980s, thousands of seal deaths were observed along the coast of the Baltic Sea, which turned out to be a consequence of a distemper virus. We now know that. But there's a couple of things that were important to note about the environment that the seals were in. And one is that the aquatic environment was heavily polluted with persistent organic pollutants and a variety of them. So there was an opportunity for these seals to be exposed. The second is that these species tend to bioaccumulate toxins due to them being higher order predators in the food chain, and they have high adipose tissue where chemicals can accumulate. So people started thinking about this connection between the ability of pollutants to make seals, in this case, more susceptible to viral infections. And there's been a lot of focus trying to understand how that might work in that space. In the 80s also, people started doing some work on research, particularly in animal models. And this middle panel shows work by Linda Birnbaum's group, where they were looking at the impact of dioxins, and one of them being TCDD, on host resistance to influenza virus. And what they showed was that the percent survival decreased when the animals were pre-exposed to the dioxins. And then the last example here is largely stemmed from epidemiological studies, which report an association between air pollution and increased respiratory infections. And I'll talk a little bit more in the talk later on about what we're learning about the relationship between air pollution and SARS-CoV-2 and COVID. But while there has been a growing body of literature regarding how pollution may influence infectious agents and vice versa, really this interface of environmental health and infectious disease and the research being done in that space is quite small. And so this was highlighted at a recent workshop in 2019, where data from a PubMed search spanning the last 10 years shows that all of the articles on environmental and health, of all the articles on environmental health and infectious disease, less than 1% really address this intersection. And so this supports the notion that fields of environmental health and infectious disease are largely siloed. And this is really despite the fact that environmental and infectious disease global health burdens are very high. We heard about that earlier related to infectious disease, but certainly the impacts of environmental health related diseases also are large as well. But there's some challenges also to merging these disciplines. And really one is that experts speak different languages across these fields. So if you think about the term exposure to a toxicologist like myself and an infectious disease microbiologist, those can have different meanings. And then certain terms that apply to one field like transmission when referring to infectious disease agents, or metabolism or biotransformation when referring to chemical toxicants, are really specific to that field. In addition, we have lots of ecosystem changes. So really taking the 3 previous talks where there was a lot of reference to ecosystem changes like land use, climate change, other drivers like even from travel, for example, now we're layering pollution within that changing system on top of all that, and how that may influence some of the outcomes that we're interested in this case, infections. So if we go back to our seal example, certain changes in climate, for example, will cause melting icebergs, which have now led to opening routes of migration for these seal populations, which potentially put certain pollutants and pathogens into close contact that may have not been there previously. We also have to think about the emergence of not only pathogens like SARS-CoV-2, but also of pollutants. And so contaminants like PFAS, which are fluorinated compounds, I'll talk a little bit about later, which are being found and detected really on a global scale and have been found at significant levels in individuals, and how that might impact their resilience to infectious is another important area to consider. And so addressing all these challenges and gaps really requires more of a systems thinking approach, such as that proposed by One Health or Planetary Health, which was mentioned in one of the previous talks. And so this is something we're really focused on at the University of Florida, and we now offer degrees at the certificate, master's, and PhD level in One Health, really trying to focus on how do we provide some cross training so we can be working at the center of this Venn diagram to solve some of these really complex problems. So to begin to understand how chemicals and microbes alter chemicals, it's sort of a two-way street. I always like to fall back on this figure, and this is a paper published by Feingold's group in 2010. The focus of this paper was to really highlight a role for infectious agents in toxicology by trying to identify common and uncommon ground between these two fields. And so this figure shows they're combining a toxicological paradigm where you have sort of on the left in the environment, the exposures, whether it's through food, water, air, soil, or dust. You have exposure to the toxicant, uptake of the toxicant, some effects observed, and then that leads to disease. So the question was, how can we sort of integrate an infectious disease model into this paradigm and really look at how it might be different to the models that just assume one agent causes one disease. And I'm not going to be able to talk through this whole model, but I did provide this paper as reading for the students. There were several challenges to this model that I think are important to know. The first is that models of infection are based on population level probabilities. The second is that the concept of risk transmission is critical to infectious disease dynamics, as toxicants generally are not being transferred through direct contact from person to person. And then lastly, infectious disease models must incorporate dynamics of growth or die-off of microbial populations. And so these are some real challenges to try to integrate into this model. And then certainly these interactions and the way that they describe them here, which I'm calling this two-way street with many intersections, can have numerous outcomes. And so you can think about infectious infections being, or toxicity for that matter being either more severe, occurring more often, the ability of a host to take a longer time to clear injury or infection, or it may lead to real novel mechanisms and responses that we haven't yet thought about or seen. So to better really define these points, let's consider for a minute what happens when a person is infected with a respiratory virus like influenza or SARS-CoV-2 in a general sense. So I want you to know on this diagram that we're working with a time scale of days to weeks. And so immediately after infection, there's this asymptomatic stage, followed in a few days with symptoms where a virus can be detected, and then followed by an immune response that typically includes an antibody production, typically IgM and IgG. And then during this time, the individual starts to recover, and in many cases can do so completely within a few weeks. And so here you can start to imagine that if parts of the normal host response were impaired or disrupted, that these curves could change shape and shift in many directions, and then the health outcome would look different. But now let's look at toxicants. And the first thing that you'll note here is the time scale, which is in minutes to hours or in weeks to years, depending on whether the exposure is acute or chronic. And so in the acute situation, this typically refers to a relatively high dose with rapid symptoms that follow sometimes within minutes. This is typical of like a poisoning situation, if you will. The chronic situation refers to lower doses of a toxicant exposure in commonly and repeatedly over time, perhaps years, which eventually lead to symptoms and noted adverse health outcomes. The third scenario, I'm not going to talk about too much today, but it's turning out to be a very important exposure scenario, and that is exposure of a fetus in utero during susceptible windows of development that years later will show in some sort of adverse outcome, despite the fact that there aren't repeated exposures throughout the lifetime of the individual. And so that's an important space that I'm not going to spend too much time on, but realize that it's certainly there and important. So in these scenarios here, you can imagine how the presence of infectious agents may alter these curves, perhaps altering the timeline to adverse health effects or lowering the bar where symptoms are detectable or observed. So much of the epidemiological data that support effects of pollution on infections is centered or anchored on respiratory infections and air pollution. And so air pollution is a term for a complex mixture that typically contains particles like particulate matter of various sizes, like 2.5 microns or 10 microns, gases like ozone and NO2, and biologics such as endotoxin or LPS, which are components of bacteria. But they certainly can contain a myriad of chemical components and metals. And the mixtures are both complex and vary greatly in composition. And in a lot of cases, air pollution is studied as a whole, or it's studied as its component parts, where associations are being looked at between, for example, PM 2.5 and respiratory virus susceptibility, or NO2 or ozone. Some of the common respiratory viruses that have been most highly investigated in this space or in the context of air pollution include the following that are listed here: rhinovirus, coronavirus, influenza virus, and of course now we have evidence to support a relationship between air pollution and SARS-CoV-2, and we'll talk about that in the next few slides here. So the epidemiological literature is rapidly growing in this space. Several studies now report a positive association between air pollution or the components of air pollution and COVID-19 related mortality as the health outcome that is being looked at. And just to give you a flavor of this, I'm just going to present one slide on a recent paper done by Eric Coker et al. And colleagues, where they examine the role of ambient particulate matter in explaining a spatial variation in deaths that occurred throughout the most extreme time period of the epidemic in Italy. And so they really went into this study with the hypothesis that higher average long-term exposure to PM 2.5 is positively associated with high death tolls in northern Italy. And just to give you a little summary of what they did, so this is a small piece of their work. But this study was focused on the area in northern Italy. And they focused on that space because there's a high level of pollution in that area based on industry that is there. And so they're also the most densely populated, and emissions in that area of the country have been monitored for years, and the air quality is fairly poor. So from their analysis here on the left, so this is now the part of northern Italy shown here, is a spatial distribution of cumulative excess deaths in sample municipalities spanning the time period from January 1st to April 30th. And then on the right, you have the spatial distribution of PM 2.5 concentration levels in the same areas, but this now spans data collected from 2015 to 2019. And so comparing these figures, one can visually appreciate a spatial coincidence between higher levels of excess mortality and higher levels of PM 2.5, particularly in these central regions, which are known to be the most heavily polluted. And so they do a lot more analysis in this paper, and really the summary of their work suggests a positive relationship between PM 2.5 concentrations and COVID-19 related excess mortality, and further suggests that a strong positive correlation between viral respiratory infection and incidence and ambient PM2 concentrations occurs with this longer term exposure to PM 2.5. So then there's a question about how is PM possibly contributing to this association, which they didn't express in this paper, but I'm going to suggest a few ways how it might today by talking a little bit about mechanisms of action. And I'm really going to focus on two areas in the next few slides. The first is related to direct interaction of, say, PM and perhaps other chemicals and viruses, and how this can modify exposure and distribution in organisms of viruses in this case. And the concept here is virus hitchhiking on PM. And the first speaker, Joe Eisenberg, actually alluded to this already in the context of environmental transport, but I'll talk a little bit more about how that might change the distribution of microbes within the lung environment. And then the second I'm going to mention is how PM or other pollutants can change the way that the immune system normally responds to a pathogen. And I'm really going to focus on the innate immune response today, which is an area where I've spent a lot of time doing research. So I'm not going to spend too much time talking about this in detail because it was already discussed a little bit in Joe's talk earlier. But the first sort of idea that I want folks to think about is this idea of virus hitchhiking using particulate matter as the agent that the viruses are hitchhiking on. And so in general, viruses are not really traveling too long of distance. We're talking about feet, and this is related to droplets and potentially some airborne transmission from one person to another. But we know that particles in the atmosphere can travel longer distances. And so there is data to support that environmental PM can be a carrier of airborne pathogens, including viruses, and potentially could change distances in which a person could be infected with virus particles. But I do think there are some questions that remain and what people are really trying to work on and figure out. And one is, viability and whether infectious particles can be transported long distances on PM. And then secondly, if there are components of, say, viruses stuck to particulates that are not maybe fully intact, when they enter the body, how does the immune system process that or see it? And I think those are questions that are still unanswered at this time. But the second sort of piece of this virus hitchhiking story, which hasn't been really alluded to yet, is pulmonary distribution. And we know that a lot of particulates can be dispersed or be distributed down into the deep lung. And that's been known for quite some time. So you can imagine if you have viruses attached to PM in this case, or even nanoparticles, which are even smaller than PM 2.5, this may promote viral translocation to the lower respiratory tract, which typically causes more severe infections. Or you can imagine the scenario where a virus is attached to larger size particulates, which might be retained in the upper airways or even in the nasal passages. And then finally, the pollutant microbe complex may alter the normal immune defenses that occurs differently than what we see to each singly. And ideas in that area could include altering recognition and clearance by the immune cells. So the body is sort of seeing this complex differently than it sees each agent singly. Or the complex could alter recognition of or expression of certain receptors. For example, the ACE2, which is the receptor that SARS-CoV-2 interacts with to actually enter and affect cells. Or to go one step further, altering how the body's normal immune defense system sees this complex. And so those are just some ideas to think about. There's lots of scenarios there, but these are some of the primary ones that I think people are interested in investigating with respect to these virus PM complexes. So I'm going to move now into what might happen with respect to the immune system, and really in particular the innate immune system. So the innate immune response to anything foreign is typically the part of the immune system that is immediate. It happens rapidly, and it's a signal really that sets up the adaptive immune arm, which is where antibodies are typically developed against foreign entities. So primary to the innate immune response are lots of events occur there, but primary is this group of receptors called pattern recognition receptors. And really their job is to act as a police force in identifying primarily microbes that are foreign to the body. And so what they do is they recognize these, they set off a series of events that are related to the production of lots of cytokines and chemokines to eventually help clear the invader, clear the infection. And so there's several families of what we call host sensors, those pattern recognition receptors. Some are which are shown here in this diagram. There are others, but for simplicity and time today, I'm just going to talk really about two. And these include toll-like receptors, or TLRs, which are shown on the left, and on the right are RLRs, which is short for retinoic acid inducible gene 1-like receptors. And what's really unique about these receptors is that they interact with specific moieties of microbes, such as viral proteins or viral DNA or RNA material. And these are called PAMPs, pathogen-associated molecular patterns. Some of these receptors are located on plasma membranes and others on cellular endosomes, and then components of the RLR cascade work through the mitochondria. These receptors are expressed on epithelial cells and immune cells, and once they're activated, they end up releasing a lot of inflammatory cytokines and interferons, and in the case of viruses, antiviral proteins. So there's sort of complex diagrams here, but the point is that you have activation up here at the top through these receptors that really lead to what I'm showing at the bottom here is the immune defense system. So just as an example, commonly toll-like receptor 4 recognizes LPS from bacteria, whereas toll-like receptors 3 and 7 and 8 interact with nucleic acid material of viruses. So in a normal situation, after exposure to virus particles, they'll eventually be recognized by these systems, which sets off this host response, if you will. Importantly, some of these receptors, and this is newer information, can be bound, activated, or blocked by pollutants. And so now this has been shown with agents like mixtures of diesel exhaust particles, ozone, and smoke. And we've done work over the past 5-6 years or so showing that new emerging contaminants can tweak these pathways as well. So I'm just going to spend just a few minutes. We've published numerous papers in this space, but we've particularly been interested in nanomaterials because they're being widely used in lots of applications like sunscreens, replacements for titanium, in computers, and lots of different areas. And really what defines nanomaterials are that they're entities that are very small. So they exist in the nano-sized range. And so with all this global use, there is some concern about the associated toxicity if individuals are exposed to them, particularly through an airborne route. So my group's been really heavily focused on carbon nanotubes, which are essentially sheets of graphene rolled up into a tubular structure. They can actually link end to end and sort of look fiber-like in some cases. And we certainly have historical information about exposure to fiber-like entities like asbestos and pulmonary disease in particular. And so we've really done some basic sort of cell culture and animal work. I'm trying to look at the ability of these to tweak these pattern recognition receptor pathways. And so in a very simple set of initial experiments, we took lung cells and exposed them to carbon nanotubes and followed with a viral infection. And this was an influenza H1N1 virus. And essentially what we saw is that we would always get a very significant increase in the viral titers, which is a measure of the number of viral progeny being produced by the cells. And this was occurring without cells being killed by the carbon nanotubes themselves. Interestingly, we don't see the same thing with other types of chemicals, which I'm not going to talk about today. But that's something very interesting, showing how different chemical exposures can be with this endpoint that we're looking at, which is viral infection. Just to summarize several papers, we've actually looked at a lot of the players in these pattern recognition pathways, so TLRs, RLRs, and endpoint genes that are controlled by those pathways, such as shown here, these interferon betas and IFIT2s, which is an antiviral protein. But essentially what you're seeing is the black bars are the normal induction you see with an influenza virus, which makes sense because this is the host defense against the virus. And as we start increasing the dose of carbon nanotubes here, you see a significant decrease in these pathways. And so we have lots of information showing how we think that is happening. But I think for this talk today, it's really just highlighting how exposure to a toxin or a contaminant can certainly have an impact on the host defense response that maybe increases susceptibility to infections. And so what do we know about this space with respect to pollution, SARS-CoV-2 and COVID-19, really with a focus on these pattern recognition receptor pathways? So what we know about SARS-CoV-2 is that it enters cells by binding to this angiotensin converting enzyme 2 receptor. And once this occurs, it ends up leading to vascular permeability, which is why a lot of folks now talk about COVID as more of a vascular disease. So with respect to air pollution, there's been just a few studies that have looked at the role that these, both the ACE2 receptors and the pattern recognition receptors play mechanistically here. So in one study, it was shown that air pollution and really in particular, NO2 and PM caused overexpression of this ACE2 receptor in respiratory cells, and this was associated with more severe COVID-like disease. Then in another study, the SARS-CoV-2 spike protein was shown to bind TLR4, and I find this very interesting because usually TLR4, as we know it, usually binds bacterial moieties and things like LPS. And so I think this is a really interesting finding, and really maybe another mechanism to consider is that certain spike proteins of viruses that may interact with some of these receptors as well. And what the result of this is that there was increased cell surface expression of this ACE2. So now here, between these two studies, and they're separate studies, we can start to maybe connect exposure to air pollution entities and alteration of normal receptors that viruses use to get into cells and these pattern recognition pathways. Another interesting study focused on the RLR pathways, and that showed that the SARS-CoV-2 M protein could interact with a protein called MAVS, which is part of this RLR system. And so MAVS is really interesting because it's actually this protein here that sticks out on the mitochondria. And once a virus is recognized by RLR receptors, which are called RIG here, they actually cluster on one end of the mitochondria. And it's this clustering or aggregation that actually continues the signaling to upregulate host defenses through this pathway. And very interestingly, we have looked at this mechanism with our carbon nanotube story and found that the carbon nanotubes actually impair this aggregation as well. So I think this is another really fascinating mechanistic area of study. And so lastly, because I don't want to focus everything on PM as the pollutant source here for looking at susceptibility to perhaps SARS-CoV-2 and other type of respiratory infections. I just want to mention PFAS. I mentioned it at the beginning. These are polyfluoroalkyl substances, so they're fluorinated compounds that have been around for quite some time, really since the 30s. They are man-made chemicals with a really interesting history. But what we know now is that we are finding PFAS really globally all over the place at significant levels because they're used for a variety of different purposes. They were used in nonstick cookware, so like Teflons, water repellent clothing, and in firefighting foam. So there's lots of uses. And we are now finding significant levels of these compounds in individuals as well. And so people are now starting to look at this compound as a potential modifier of the human immune system. And so that begs the question as to whether it would affect the immune system in such a way that people would be more susceptible to infections. And so there's been a couple studies out now. Grand Jean's group really has done a lot of this work. And just to give you a little example or flavor of this, they recently published a paper looking at 323 COVID patients, and those which had very high levels of one type of PFAS compound, which is PFBA. And they were really interested in this compound because it tends to accumulate in the lung. And what they found in that study was that those individuals that had high levels were more than twice as likely to develop severe forms of COVID and had higher chance of being hospitalized, entering intensive care, and end up dying. And so there's quite a few other studies out there that are linking PFAS exposures, or at least PFAS levels, high levels in serum and other tissues, to immune effects. And so it's certainly relevant to look at with COVID-19 and SARS-CoV-2 like infections. And in a separate study by the same group, and this was not COVID focused, but interesting and something to think about, is they looked at antibody responses to tetanus and diphtheria vaccines and showed that the response to the vaccine was weaker in children who had high levels of PFAS. So whether that is the case as well with COVID, I think we don't know. I haven't seen anybody do those studies yet in the same capacity. And so I'm going to end here by just talking about a few gaps and opportunities. And I think the first point is that really toxic and pathogen interactions are not that well investigated. And I alluded to that at the beginning of my talk. So I think there's a real need for this cross-disciplinary and system approaches. So one of the last speakers talked about the real need to better integrate ecology and epidemiology. And I would say we have to do that in sort of the tox, microbiology, and infectious disease space as well. So we have a better understanding of these exposures and what they mean from a health standpoint. Also considering that I think there's a lot of novel outcomes that we haven't yet thought about. I think the second point here is resilience to infections are intimately linked with our immune system. And so really more work on understanding how our immune system normally responds and how that might be perturbed by pollutants is really largely unknown. And I think this is important because understanding mechanisms clearly can lead to us developing things like better biomarkers of exposure, better diagnosis and treatment for some of these diseases, and certainly better public health interventions. And then lastly, I want to make the point that identifying which pollutants and agents are priorities is important. And along those lines, generating baseline surveillance data to support epidemiological-based assessments is needed, including a focus on vulnerable populations. So we are trying to do this kind of work, for example, in agricultural workers, where we're looking at PM exposure and respiratory infection susceptibility. And we are really starting with very limited information, for example, on respiratory infection status in this community, despite the fact that they can have fairly high levels of exposure to particulates. And so that's some of the challenge that we've run into in trying to do some of this work in these populations. And I'm going to end here with a picture that is supposed to set a little bit of feeling of wellness here, because I do that as part of my job as well. And so this is an unscripted picture of my daughter. And if we could all take a few minutes to feel like this every day, I think it would do us some good. So I will stop there, and my contact information is here, and I'm glad to answer any questions.
Thank you so much, Tara, for that excellent talk. Very, very fascinating. There are several questions in the chat, and I'll just pull some out here. One question was about the hitchhiking mechanism of viruses hitchhiking on PM, and does that relate only to short-term air pollution exposure, or also is it relevant for long-term exposure?
I think if I understand the question directly, the question might relate to how long can virus particles be maintained on PM. And I'm certainly not an atmospheric transport expert, but I think the question there is really, are we thinking about infectious particles or bits of particles? And so I think the answer might be different depending on which one of those we're talking about. I'm not sure there's a lot of evidence to support that infectious particles might stay on PM for very long periods of time. But what is a challenge there is our methodology to be able to really understand how infectious particles might be that are attached. Being able to capture infectious particles out of the air is challenging enough, and then having them attached to PM, I think, is a whole another challenge. And part of that has to do with the methods that we have to know whether virus particles are there. How do you know without destroying the sample, right? So that's an interesting area where we need more work. Now, for pieces pathogens or pieces of microbes, perhaps those could stick around a little bit longer. And so there's a potential there for the immune system to respond differently to a complex that has pieces of, perhaps viruses on them versus the whole infectious piece. So, yeah, I think there's a lot of work to do in that space.
Very interesting. And then there was another question about the possibility of viruses attached to PM influencing how far the PM is able to deposit in the respiratory tract.
Yeah, I think it's both ways. So, I think particulates could potentially drag, if you will, viruses lower in the respiratory system, but I think having viruses attach, it could also influence where the particles go as well. So, we talk about this a lot as particles influencing viruses, but I think that's definitely a two-way street for sure. I didn't talk about it, but there's clearly other defense, systems like mucociliary, escalators and all sorts of things in upper airways. And certainly depending on both, the size and the properties of these things, it will dictate where it goes in the lung. I mean, there's a whole science of inhalation of particles and impaction and angles and, how they move their way through the airways, and certainly having something attached to that will change that for sure.
So there's another question here which sort of gets at the sort of exposome concept here, which is, of course, we're not exposed to just one chemical at a time, but complex mixtures of chemicals. And how would that complicate our understanding of the true cumulative impact of pollution on susceptibility to infection?
Yeah, I wish I had a good answer for that, because I've been a toxicologist and really only working in the infection space about the past 10 years, but about 20 years in toxicology. And just within the field of toxicology, we completely struggle with the idea of complex mixtures. And certainly how to piece apart, what compounds are doing individually versus what they might do together. So certainly there's a need for that space, but it's the exposome, like you said, so you have to take all those things into consideration. So, perhaps with the development of maybe my hope with movement in AI and computational space, that, maybe we can get a better handle on, linking mixtures and biomarkers and effects, biomarkers of exposure and effects, so that we better understand how those work internally. But it's a really complex equation. Even ToxCast databases, for example, which is a very large database where we're putting all sorts of information about biological activity of chemicals, but it's still looking at chemicals one by one, not in a mixture form. So I think we have a ton of work to do in that space, and then certainly how all that together impacts the immune system is complex. So I don't have a great answer for you as to how we do that, but those are just some of my thoughts on it.
And mainly as maybe this is the final question for this session, but it's a sort of broad question about how, in your view, toxicologists and epidemiologists can benefit from working together more effectively, and maybe you can comment on that, particularly in the context of COVID.
Yeah, part of it gets back to this sort of more systems thinking approach. I think we've been doing that here. So I mentioned Dr. Coker's work, and he's in my department, and so we inherently work well together because we're in the same space. But I do think we have this issue of understanding each other's language. And I know it sounds like a simple thing, but it actually is not a simple thing. And I'm really living in that space right now, because I'm teaching a course that is called Mechanisms of Environmental Disease. And we're covering pathogens and chemicals in the same course, which is really weird. It's a weird thing to do. And what we're finding is that communication across these disciplines is a major barrier. So I think we have to start there, especially with, training students that are coming through the pipeline here, is how do we better talk across these disciplines? Because you can't get training in every discipline, right? But you can communicate better across them. And so I think that's really an area that we need to pay more attention to. And I think it's overlooked because it sounds simplistic, but I don't think it is.
Yeah, I think anyone who's tried to do sort of multi or transdisciplinary work knows that it's most definitely not a simple thing. So thank you so much to Tara and our other speakers. It's been a really interesting session, I think, today. Just to mention that the learning activity for today is posted on Moodle along with the suggested reading, and it will be open until Sunday. We know if some of you have had a bit of difficulty accessing the learning activities in Moodle, so they'll be open for some time. So please go there and complete the learning activity before Sunday, and you should have received an email from this website, or sorry, this address, and that should have your link to the Moodle system. And if you have any problem, you can reach out to Ariadna at this email there. And with that, I will just say thanks again to the speakers and to all of you for the great discussion in the chat, and we'll see you next Tuesday.